Hi, Mindsetters, and welcome to your favorite Learning Extra show. It is, of course, Mindset Learning Extra, and in studio today, it is, of course, me, Pumi, joined by our teacher. How are you doing, sir?
How are you, Pumi?
I'm good, thanks. How are you?
I'm just managing.
You're just managing.
Yeah.
It's a beautiful Wednesday afternoon, and today we have the Mindsetters joining us in studio.
That's correct. We got 6 of them today.
Oh, nice. Hey, guys. How you guys doing? Oh, okay, cool. We're good. Which school are they from?
All from Amatimo.
Amatimo, can see they're not really using the mics, but it's okay. They're from Amatino. Guys, please make sure that you join us today. It is an absolutely amazing show. Log on to our Facebook page in order for you to participate on our show. It is www.facebook.com/Learn Extra, www.facebook.com/Learn Extra. You can also tweet us right there on our Twitter account. It is @Learn Extra. Follow us. We'll probably follow you back. Okay, we'll follow you back. All right, then, let's just get straight to learning.
Thank you so much, Pumi. Guys, today, welcome Mindsetters. We're busy with Prelim Paper 2 questions, and we're going to go through some of them. Let's see what we're talking about today. All right, we said that already. We're revising papers 2 of the prep exam from a few provinces. First up is the challenge question, and our guests in the studio, I want you to note this because later we're going to ask you to help answer the question. They say in a certain species of plant, and people, this is a real challenge question. If you are not a pro in genetics, then the dihybrid, you rather leave it slightly out. But if you are doing well with genetics, then you should try the dihybrid. Like I said, if you are slow on genetics and you are one of those that are struggling, then rather focus on the monohybrid crosses because that's where the bulk of the work is. In a certain species of plant, Acer rubrum, red maple, tallness is dominant. Homework, remember, tallness is dominant over shortness, big T, small t, and red fruit color is dominant over yellow fruit color. You can see already we're talking of 2 characteristics, so therefore it's a dihybrid cross. A plant that is homozygous for both tallness and red fruit, and you've got to go back there to see what is dominant, is crossed with a plant homozygous for shortness and yellow fruit. List the phenotypes and genotypes of the P1 generation, the parents, and the F1 generation, the offspring. Now, if the F1 generation in question A, there, was interbred, in other words, this F1 generation here was interbred, what are the chances of the offspring being short with yellow fruit? Remember, you need to figure out what is dominant, what is recessive, and remember, dihybrid crossing is based on the principle of independent assortment. That means the tallness or the shortness does not affect the red and the white. They segregate separately, independently from each other. So keep that in mind. That's where we're starting with. Remember, we don't have summary, so we go straight into the questions today, and we go into a question from Gauteng Prep 2014, so you youngsters should be au fait with it. The first question, remember guys, John has explained to you, that's team 1 there, and this is team 2. Team 1 on that side, team 2 on this side. Every time you answer a correct question, you're getting 1 point. John is keeping the score, okay? If the viewers ask us a question and you answer correctly, you get 5 points. Fortunately, there are no deductions for wrong answers. So there's no negative marking, all right? So I want both types of answers because remember, wrong and right answers both help us to learn. Since 1972, biologists have studied finch population, homework, finch populations in the Galapagos Archipelago Islands. The graph below shows the change to the number of finches, the rainfall, and the number of small seeds. So there's 3 things in this graph. Over a period of 7 years on one island, that is the Daphne Major Island, small seeds, another clue, are abundant in rainy seasons, and large seeds are drought resistant. In other words, when the rainfall is low, there will be more large seeds than small seeds. That is the information that you had to pick up in the intro. And that same information is packed into this graph now that we had to read. Rainfall is the solid line with that oval shape. The dotted line with the cross on it is the number of inches, and the dotted line with a little triangle is the number of small seeds per square meter. So we got 3 bits of information in this graph. Important mindset is that you read the whole question and you understand what is said in this graph. In other words, you need to follow the lines and see what is happening there. So let's see what we can read from this graph. We're starting in 1974 and moving to 1980 firstly. And on the y-axis, they're telling us the amount of rainfall, the number of inches, and the number of small seeds based on this key that is here on this side. So independent variable, dependent variables. There are 3 dependent variables in this question. First question is, and we're going to ask team 1, in which year, and unfortunately guys, remember the easy question may fall on one team and so on, but it will balance out. In which year was the finch population the highest? In which year was the finch population the highest?
1975.
Fatima says 1975. Let's see. Dong, dong, dong. Oh, she's correct. That is 1 point to team 1. Briefly describe team 2. Haha, shame. Briefly describe the trend. John, you should give them 2 points for this one because it's a more difficult question. Briefly describe the trend. Let's see if they get it right. In the finch population. Oopsie. In the finch population from 1977 to 1980. Okay? Are you ready? Yeah? Let us look at it. Let's keep the graph there, right? The finch population, remember the finches are the dotted line with the crosses from 1977 to 1980. Yes?
From 1977 to 1978, it increased.
Right, it increased first. There's the increase. Mindsetters, watch that. Increase, yes?
And from 1978 to 1980, it decreased.
It decreases. Notice the answer was given with specific dates. So that's correct. The answer is correct. You can't just say it increased and then it decreased, or it increases and decreases. You must say from 1977 to 1978, specifically it increased and thereafter it decreased. Okay, there we go. That is correct. Great, we're doing well, guys. Name the environmental factor, team 1 now. Name the environmental factor that caused the changes in the finch population described in question 1.1.2. Just the environmental factor, Wasim?
Rainfall.
Rainfall is correct. They're saying amount of rainfall is not really necessary to say amount because they asked for the environmental factor. So it's rainfall is good enough. Well done, Wasim. All right. Ah, shame. I'm going to have to change it somewhere along the line for me. These guys are getting the longer questions and those guys are getting the one
Yeah,
answers.
they're taking
So
all of the
again, yeah, if
answers.
they give us the answer, they have to get 2 points. Eh? Okay. Use Darwin's theory of natural selection to explain how seed-eating finches, listen very carefully, with large beaks evolved on Daphne Major Island over the period 1977 to 1980. Let's go back to our graph. Arshad?
There was variation in the beak size of the finch population.
All right, let's start. Let's just jot some of your points so we know whether you are with us or not. He says there was variation in the finch beak size, yeah?
Right? Then some had large beaks,
Large
others had small beaks.
And small. Excellent, yeah. Talk clear and loud, right? Don't forget.
During the season of 1977 to 1980,
1977 to 80,
there was low rainfall, drought.
low rainfall. Excellent. Well captured, right?
Abundance of large seeds, so there was a lack of small seeds.
So there was abundance
Of large seeds.
of large seeds. Good.
So the finches with small beaks were eliminated.
Small beak finches eliminated.
Right? Finches
Good.
with large beaks therefore survived.
Large beaks finches survived. Yeah.
The characteristics of the large beaks were passed on
Characteristics. When was this passed on?
over several generations.
Yeah,
in
when?
future generation.
When they did what? Characteristics were passed on.
During reproduction.
Good. They reproduced. They reproduced. Because they survived, they could reproduce and therefore passed on the gene which held the characteristic, the favorable characteristic of large beak. I give you that point. That's good enough for me. That's the bulk of the answer. Let's just put it into a little more formal setting. So there was variation in the beak size, and viewers, you can see whether Arshad has given us the correct answer as well. There was variation in the beak size of the finch population. Some finches had small, others large. During the season 1970 to 80, low rainfall or drought, abundance of large seeds, or the lack of small seeds. The finches with small beaks were eliminated, large beaks survived, and characteristics of large beak were passed on to offspring, increasing the proportion of large beaks in future generations. Excellent. Good. 1.2. And we come to group 1 again. The graphs below show the breeding seasons of two species of monarch butterflies that are phenotypically identical. Listen to the question. Phenotypically identical, live in the same habitat, and feed on nectar from flowers. But they are two species. Remember that. Study the diagram and answer the questions that follow. Species A, reproduction time in months from January to May. Species B, from June to November. That is the breeding seasons. Sometimes different but related species do interbreed. Use the information provided to explain how the two species above are prevented. They are similar, but how are they prevented according to the information only? What prevents them from interbreeding?
They reproduce or mate during different times of the year.
Different times of the year or different breeding seasons. Excellent, your answer is correct. Breeding season is different. Name two reproductive, group 2. Name two reproductive isolating mechanisms other than the one mentioned here, other than breeding season. Uzma?
Different courtship behavior.
Different courtship behavior, yeah?
And reproductive organs not compatible.
Uncompatible reproductive organs. Good, we will accept those two. Remember, there are others, and the exam guideline specifies different courtship behavior, adaptation to different pollinators if it's plants, infertile offspring or hybrid sterility, that's the other one. And the other one is, the fourth one is the one we spoke on top, different breeding seasons. Remember, mindsetters, for your exams, you stick to those four. Different breeding seasons, different courtship behavior, different pollinating agents, and hybrid sterility or infertile offspring. Remember, here they only ask you to state it. In an exam, they can ask you to explain it, each one. And it's easy to explain. I'm not going to go into the details of that. Good. 1.3, an investigation was carried out to determine the effect of antibiotic on two types of bacteria that cause TB. The diagram below shows the result of the antibiotic after day 1 and 2 on a 5-day course to treat the infection. The bacteria were cultured in a petri dish at a temperature of 37.5 degrees. So before the administration, there's bacteria A and there's bacteria B, the ones with the dot. After administration, you can see the difference in population. Day 2, this is what's happening, and that is what we're getting in the end there. Suggest one hypothesis for this investigation. Where are we? We're with you guys. Ah, now you're getting another one. Hypothesis for this investigation? Umar, you haven't given us anything yet. Are you shy from the mic? Fatima, help you.
Bacteria B are resistant from the antibiotic.
Bacteria B are resistant to the antibiotic. I don't know whether to give that point or not. We're going to hold back on that particular point because, first of all, we are saying in the present tense, we must say the hypothesis in the future tense. So what we are saying is we're doing this investigation. What's going to happen in the future?
Okay.
And with that, We're going to go for a short break. We catch you mindsetters just after this break. Our contestants are neck on neck. They're almost equal on points. So let's give them a break.
Absolutely, absolutely. Now, before we take a quick break, mindsetters, let me quickly remind you about the competition that we're running. We are giving away 4 Chevy Sparks. So if you want to be that lucky winner and proud owner of one, make sure that you enter the Be a Bright Spark competition by simply buying yourself a Safeway K53 book from any outlet around, any outlet or bookstore, and then also you can buy and download it from from your Samsung Galaxy app, and then from there on, keep the receipt. Do not also forget to share the page right there on your Facebook page so that your friends can also get to enter the competition. And once you did that, remember that once you like the page, you can't just be given the an entry point. So all what you need to do is just to buy it. After buying the app, enter, or rather like the page, and after liking the page, you buy the application. It's just as simple as that, mindset. It's not really as difficult. Make sure that you log on to our Facebook page because more details are attached to it. Like the Safeway K53 page right there on Facebook as well for all of that and more. Remember, there's a question that I've asked you guys earlier on about people that buy their licenses, of which I don't really find it interesting. So please, guys, give us your views. Tell us what you think about it. Do you know of anyone who buys their licenses? Please make sure that you share all of those views right there on the Safeways K53 Facebook page. See you after the quick break. Welcome back from the short ad break, Mindsetters. We are still doing life science with Aslam and the Mindsetters joining us in studios. Guys, are you still good? Okay, which team is winning?
John will have to tell us.
Okay. You guys need to keep up, hey? Alrighty, cool, Aslam.
Thank you, Pumi. Right, so we were busy with that hypothesis there. Notice that there are many alternatives here. And notice, mindsetters, we're starting there with the independent variable, which is antibiotic. The addition of the antibiotic is the independent variable. One is, we're saying that antibiotic treatment will have no effect on the bacteria. It's a hypothesis. We're not looking at the result. Remember, hypothesis got nothing to do with the result. The hypothesis before your experiment. Or we can say that antibiotic treatment will cause population size of the TB bacteria to decrease. Or we can say the same thing will cause the population size of the bacteria to increase. So the two variables were administration of the antibiotic and the bacteria on the other side. So those are the variables must be in your answer. So obviously the next question is now blanked because I've already told you that. So which factors in the above investigation is the independent variable? That would be the administration of antibiotic, and the dependent variable would be the population size of the TB bacteria. Okay, so those ones they are struck off. Now we come to group 2 again. Which bacteria, A or B, is resistant to the antibiotic?
B.
The answer is B. Let's see. That's correct. So we will give you that point. The answer is B. Good. Explain the effect, group 1, that the antibiotic had on the bacteria after the first and second day of treatment. What happened?
The bacteria.
You talk louder than Lucky.
The bacteria started becoming resistant to the antibiotic.
Which bacteria became resistant?
B.
All right. So listen very carefully when we explain that. We will give you the point for that. But remember, mindsetters, that when we say become resistant, although it's accepted that organisms become resistant, but it's not the organism itself, it's the population that becomes resistant. Remember that. That when we look at this, we had 2 types of bacteria. And what had happened here is the one type, this one here, A, seems to be eliminated as we move on. and B seems to thrive and get more. So in other words, B was already resistant before the antibiotic. I'm hoping you understand what I'm saying, guys. That you either have the factor or you don't have it. So what happened here is, remember when we explain natural selection, we say variation. This is the variation here. Some were resistant and some were not. And we're going to talk about that just now, and we're not going to ask that question. We're going to carry on with that. I'll come back to that now. So we're giving you that point there, that all the non-resistant bacteria or bacteria A were killed or destroyed, and the population of size of bacteria B increased. Name the process that leads to the difference in the population size of bacteria A and B.
Natural selection.
Natural selection is correct. So there you get your point. Predict the consequence. Group 1. Should a person fail to complete the antibiotic course?
Too many resistant bacteria.
There will be lots of more resistant bacteria, and what's going to happen to the health of the person?
He will die. We won't be able to resist.
They won't be able to resist. Okay. We'll give you the point for saying that there will be lots of resistant bacteria multiplying. And the second part of the question I asked you is that means the person who is sick is going to get more sick, or he'll remain sick. Well, it doesn't mean he's going to die, because we can still go to hospital. Remember that. We can still take him to the hospital or whatever. Okay? So the severity of the infection will increase. All right. Why must the bacteria be cultured at 37.5 degrees, Uzma?
To suit human temperature.
Why? Because where were they taken from?
From humans.
From humans. These bacteria grow in humans, so therefore they're shooting that. We'll give you a point for that one there, because bacteria are taken from human, and 37 degrees is optimum temperature for this particular bacteria. State 2 precautions that must be taken to ensure that the results of this investigation are valid. So this is an open question to both groups now. 2 precautions that must be taken to ensure that the results of the investigation are valid. Group 2?
It must be kept at the temperature 37.
Good. First of all, it must be kept at 37.5 degrees. That's going to be one of them. So that's one. Good. What else? Anything else? Anybody else? Going once, going twice?
Use the same antibiotic.
Good. You must use the same antibiotic. And we can add to that we start with equal, if we can, numbers of each bacteria as well. We can't start with 10 of the one and 2 of the other one and expect that. You wanted to say something?
So can we also say equal doses of the antibiotic?
Good. Also, yes, use the same antibiotic and the same dose of the antibiotic. There are 2 different points. One is using the same type of antibiotic, and the other one is the same dose. Excellent, guys. Well done. Okay, we move on to question 2. Still on evolution, but evolution humans. Diagram below represents the skulls of 2 organisms, a modern human and a gorilla. Study the diagrams which are drawn to scale in this case, and answer the questions that follow. Right, they're giving you A and B, and we start with group 1. Which diagram, A or B, represents the skull of a gorilla? If you get this wrong, I come there, I kick you, and on TV we show you how we can kick with our size 7s.
B.
It's B. You're so lucky.
B.
Good. It is B, so you get your point. It's obvious. That one, mindset is, if you get this wrong, there's something wrong with you, because that means you can't identify yourself from here. You don't know which skull you are looking at in the mirror. So you must be careful. Good. Which organism, A or B, group 2, is bipedal for most of its life? Notice they use the word for most of its adult life.
A.
A is correct. It is humans. Remember, when we are comparing primates, we use the similarities and the differences. Under the similarities, we say that primates all have the capability or the ability to be bipedal. But under the difference, we say that humans are always bipedal. Okay, so there's a similarity and a difference. So guys, learn that one, because you can use it for similarity and you can use it for difference. The same as size of the cranium compared to body size. we say that all primates have a larger cranium. But under the difference, we say humans have larger cranium than other primates. Good. That one is correct there. Explain 3 possible advantages of bipedalism for an organism. Group 1.
Balance.
Wasim.
Balance, further sighted, hence free to do things or hold things.
Good. Okay, you've given me enough in that I need from there. One is ability to scout the environment better. Okay. Now notice they use the word. What did they use? Explain here. So notice here we said first of all ability to survey the surroundings better. So that's the advantage. But explaining it why? To see where the dangers are. Other one is fast movement, so that they can get away from danger also. Freedom of the forearm and hand for using for other reasons. And they're exposed to less UV light for temperature regulation. You can also say that the body is exposing more also, so that it can lose also more temperature in that way. And I'm surprised Lucky didn't tell us about this one. Display of the male genitals to attract a mate. Okay? That's also, especially the gorillas do that. Not all organisms do that. Good. We are now looking at still evolution, but we're looking at a phylogenetic tree now. And guys, once you learn how to do one phylogenetic tree, all others become easy. You just need to get to the bottom of it to see where they meet, etc., etc., etc. Okay, so here we have 15 million years ago, we had the orangutan coming there. From there, the gorilla coming from the same line, but further away. Then at 5 million years, we get the chimpanzee. From the chimpanzee branch, we get the bonobo. And continuing down that line, we get the human, according to the principles of evolution. So we were with which group? This group 2. Which species is most closely related to the chimpanzees according to this particular model?
The bonobo.
The bonobo is correct. You get your point for that one. Good. Excellent. Group 2. How long ago did the common ancestor of gorillas, chimpanzees and humans arise? Gorillas, chimpanzees and humans.
15 million years ago.
Is that correct, Lucky? You're not sure. Group 2, you want to take a chance? Gorilla, chimpanzee and humans.
7 million years
7 million years ago is the correct answer. Remember, there's it there. 7 million. That's the common ancestor. But, all right, let's see. Let's see who's right. That's we are right. Okay, good. Okay. Which species is most, back to group 1 now. Which species is most distantly related to humans?
The orangutan.
Orangutan, and you'll choose the one with the R in it to give us an answer, Lucky, with your special R's. Okay, orangutan, okay? Right. Group 2, explain why the following statement is not true. Humans evolved from chimpanzees. And again, you have to look at the diagram.
Humans didn't evolve from chimpanzees, but the common ancestor was the same.
They had the same common ancestor coming down this line, and they not coming, if they arose from chimpanzees, they would have had to come from this line. They're not coming from this line, but they share a common ancestor. Excellent answer. Okay? They are in the same, they have a common ancestor, but not in the same line of development. Good. Now we're moving on to genetics. We've done a lot of evolution, guys. Now we're moving to genetics. And here it's going to be a little bit more complicated because you're going to help me to build this model or this format, and only if you get the whole thing right, you're going to get your points. Let's see how we go through this. In mice, the allele for red fur color, R, is dominant over the allele for gray fur color. It also happens that all embryos which are homozygous for the dominant factor, that will be RR, will always die before birth. If a colony of heterozygous mice were inbred to produce 300 living mice, heterozygous would be what? Two small R's. Heterozygous.
One big, one small.
One big and one small. Yes? Because heterozygous means that the alleles are different. They're not identical. Huh? How many of these offspring would be likely to have red fur and how many will have gray fur? Out of how many? 300 young living mice. And remember what they said here. Here. Will always die before birth. Can you see the homework we have to do before we start even answering this question? Okay? Use a genetic diagram to show how you arrived at your answer. So, we are saying two heterozygous mice, isn't it? So, phenotype, first of all, what color are they going to be? What is dominant? What was dominant? A red fur color, isn't it? So, if they were heterozygous, that means both would be red. Are we agreeing with that? All right. Give me the genotype. I think I'm going to take you 1 point at a time, and then you can get your points. What is the genotype of both of these? Because they told you it's heterozygous.
Big R and small r.
Big R, small r.
Big R, small r.
Good R, small r. Excellent. Right. Gametes for this particular gametes for these things here. What do we do?
Just separate
The separate these
spaces in
guys
between
into
R's.
R and small r, capital R and small r. That's all we do there. Okay? We can do it there or we can do it there. R, small r, small r. Good? Okay, both groups now help me.
Capital R.
Capital R, capital R. Capital R, small r.
Good,
Capital R, small r.
good.
Small r, small r.
And if I wrote this like that, would it be wrong?
No.
No, it's not wrong because it's the same thing there. Excellent. So now the problem is, of these now, what's the answer to the question? How many will have red and how many will have gray? Remember, there's 300 of them. There's 300 of them. How many will have red and how many will have gray? But remember the rider earlier. So of this offspring, what will represent 100%?
R, R.
Which one? Yeah, you see what she's doing? He's trying to confuse me. Eh? He's trying to hit me with a curved ball, RR. RR, they all RR.
Capital R, R.
This one?
Yeah.
No. What did they tell you about this one?
All dead.
They're all going to die. So we left with these lot. So this is equal to
300.
300. Out of these 300, how many are gray? Arshad, the maths guy, let's hear. How many
100.
are gray? Remember, gray is recessive.
What's that? So, yeah. 100.
Why 100?
That's
Because
small r, small r.
1/3 of 300 equals
100.
100. And how many will be red?
200.
200 obviously, because 2/3 are there. Notice the thing, the catch in the end was only that these guys died, so you're left with these guys, and these guys represent the total of 300. And we got 3 of them, 1/3, 2/3. Are we clear with that one? Can you see how they can just change the question a little bit and confuse you? Okay, that's the answer we've just done now. We've done that already. And that's our answer there. That one there, and we have got our answer there of that particular case. Excellent, guys. Let's move on. 135. Blood groups in humans are controlled by multiple alleles. As a result, there are 4 possible groups: A, AB, B, and O. Study the graph below, which shows the percentage, and answer the questions that follow. So they're giving you the blood groups here: O, A, B, AB. And they're giving you percentage of people in the sample that have the different blood groups. Okay, are we clear with that one? Happy? Explain what is meant by multiple alleles. Fatima?
There are many different alleles.
There are more than 2 alleles, so many different is correct. We'll give you that there. There are more than 2 alleles possible at one locus. And remember what we're saying here, we are saying that for blood group, we can have IA, which is one of a possibility. We can have IB, which is a possibility. And we have small I as a possibility. capital I with a superscript capital A and capital I with a superscript capital B denotes that these two are, what are they? Why are we using capital letters?
Dominant.
They're both dominant. Whereas this one here is recessive. Just an added explanation there. Which blood group, group 2, is the least common in the human population according to this graph?
Group AB.
Blood group AB, excellent. That didn't need rocket science. Okay, it's straightforward. Recent population statistics show that KwaZulu-Natal has a human population of approximately 9.2 million. Oopsie, I knew I'm going to do that sooner or later. 9.2 million, okay? Oops. Calculate the number of people who will have blood group O in KwaZulu-Natal. Let's look at blood group O. What is the percentage? What's the percentage?
49.
49. So if you have to work this out, what are we going to do? 9.2 million
Times
times
49 over 100.
49 over 100. Okay, and we're going to use the calculator to get our answer, and the answer is 4,508,000 people. Is that correct? I'll check on the calculator. I'm using my head, or rather, I had the memo. Okay. I'm hoping mindset is that we saw that, and I just turned around and I saw the time as well. For me, it's your time.
Absolutely. All right then, guys. Last week we asked you a very interesting question, last week Thursday, and the question was posted on our Facebook page, and the question was, what is your belief in the cause of unnecessary road fatalities? And interesting beliefs came through. And guess what, guys? We now have a video on the Safe Ways K53 Facebook page and also the Mindset Learn Extra page where you can get to see that video. And one more interesting thing is we had a question from Tabo who wants to know if you can get the K53 application if you don't have a Samsung device. Guess what, guys? You do not only have to have, if you don't have the Samsung application or other device to download the K53 Safe Ways, make sure that you go onto the Google Play, it's the Google Play Store. There's actually a very interesting or rather special discount running. So make sure that you don't just limit yourself because you don't have that Samsung device. You can also use the Google Play Store to buy and download the K53 Safe Ways. Right now, guys, let's take a quick break, and when we come back, it is, of course, more learning and learning extra. Welcome back from that ad break, guys. You know, here at Mindset, we just love giving you guys prizes and gifts, and you know, we just have a whole lot of competitions. So please take advantage of those competitions, because right now we are having this one competition that is closing tomorrow at 10 to 6. It is the Get Connected competition. And I'm sure that you guys have been trying to vote for your favorite teacher right there. I have my teachers that I have teachers that I'm thinking of voting for. So please make sure that you are voting for your favorite teacher. It is simple as you going to our Facebook page, logging there and checking out what you need to do in order for you to get yourself winning. You could be winning yourself a Sony Xperia or a time from Vodacom. Aslam, I think I'm going to vote for you.
I hope so.
You know,
Otherwise you're in
I
big
have
trouble.
to. Actually, I'm at it right now.
Okay, thank you so much. And we better tell those guys too.
Yeah, hey,
When
you
they
guys
get
have
home, they're
to
going to
vote.
vote for me, otherwise they're not going home.
Yeah, hey.
I've got to buy some votes today. Guys, we're starting with a challenge question, so just so that we don't get caught up in the end that we don't get there. So let's do the challenge question first, and then we had one or two questions that we'll take from you.
Okay.
The one I remember, I think, in my head, but we'll check it up. Just
Okay, well,
refresh
I
there
will
so long.
just- okay.
So here's your chances again, guys. We're going to start with group 1 again. A plant that is homozygous, all that I'm not reading again. What is the, what is it? They want the phenotypes and genotypes of the P1 generation. So the P1 generation, they're looking at a homozygous for both tallness and red fruit color is crossed to the plant, homozygous for shortness and yellow fruit. So obviously the phenotype is given to you. tall, red, and short, yellow. I want the genotype. For the first one, tall and red,
Capital T, capital T.
capital T, and red,
Capital R, capital R.
excellent, capital R, capital R. And the other side, to get the full point, if you got this one, the other one's easy.
Small t, small t.
Small t, small t,
Small r, small,
small r. You get the point. Excellent, good. So that's the story there. Remember, mindset, some of you make the mistake of writing it like this. You write TR, TR. Now you are mixing tallness with red. It doesn't work like that. Imagine if eye color was mixed with bum size. What would have happened to some of you? Okay, so let's not get that way. Okay, now we come to this group here. What about the F1 generation? In other words, if we made all these guys with these guys, all the offspring will be the same. What will they be?
Capital T, small t,
Capital T, small t,
and capital R, small r.
capital R, small r. Excellent. You guys know dihybrid. That's excellent. Good. That's all I wanted to know there. And they'll all be tall and red because there's a capital there and there's a capital there. Both dominant genes are there, so when the cat is away, the mice will play. There is the cat, so mice can't play. Good. We move on. If this F1 generation, now this is the tricky one, group 1. Let's hope you know what you're talking about. If this F1 generation is interbred with each other, what were the chances of offspring being short with yellow fruit?
Is it 8 over 16?
8 over 16 is incorrect. You want to take a shot? It's absolutely easy. It's your chance to get one more ahead. And I'm only giving you 1 second to do that because we need to move on.
1 over 16.
1 over 16. It's short. Let's go back to dominance. It is short. It's recessive. Good. And yellow is also recessive. Excellent. It's 1 out of 16. So you go 1 extra ahead. Good. It is 1 out of 16. Or 6%, you can say. Remember, guys, group 1, your mistake. Remember, when we have this cross, we have the phenotype ratio 9:3:3:1. This 1 is recessive. There, that's the one we were talking about now. Because we spoke about short and yellow. This 3 is dominant recessive. That means tall with yellow. This one is recessive dominant. It could be either one of them. So here you'll have short with red. And this is dominant. That means tall and red. These are the 9. And the 3 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 and the 1. Good. Excellent. Right. You got a question there for me?
The question here is coming from Spamandla Mfundo Shenge, wanting to know, help me here, please describe how each of the following contributes to variation. 1, the crossover and the random mating.
They want to know how the following
Yes.
will lead to variation. 2 things. What these 2 things are? They are crossing over and
random mating.
random mating. Don't confuse it with the other story. Right. Which group wants to take the shot? Fatima? Lucky?
Crossing over will result in hereditary information being switched.
Meaning?
Cause
Where does this take place, this
variation.
crossing over?
In the first prophase
Prophase 1
in meiosis.
of meiosis. Right, good. What happens?
Hereditary information is swapped
There's
from chromosome.
swapping, nice simple word, or exchange of hereditary or genetic material. Excellent. For that one there, you get 5 points, that group, by the way. You want to take a chance with the second one, random mating? Random mating. You want to go for that? You can go another 5 points, Fatima.
During mating, it is like every species has different genetic information.
Right.
So there is there's like a vast majority of, like a lot of different genetic information. So
Yeah.
it's possible that any any organism can mate with another organism, obviously of the same species. So that results in variation.
Group 2, you're in trouble. All right, that's the answer. That's the 5 points for you as well. What Fatima is saying, she said it right at the end, though. She had a long story before that which wasn't relevant, but she gave the answer in the end, that within a population, there's no guarantee which particular one is going to mate with the other one. So it's a random. Random means there's chance. Anyone can mate with anybody. And those two have their own variety within them, and that brings about variety. Excellent. Let's move on. Hey, I don't know how this group is going to come back, but anyway, we go to group 2 now. According to this one here now, if you know this one already, you're going to get 1 point at least. Study the genotypes and phenotypes below that show the alleles of color blindness are inherited. X and Y represents sex chromosomes. Carriers do not suffer from color blindness, that's a clue, but can pass the allele for color blindness onto their children. They give you a whole list there. Is color blindness caused by a dominant or recessive allele?
Dominant allele.
Dominant is incorrect. And obviously I can't pass it on to you because then it's obviously the next answer, and that is recessive. I mentioned, Arshad, you weren't listening when I said that carriers do not suffer. That was the clue. If a carrier doesn't suffer, it means it's caused by a recessive gene. Okay? Explain why Sam is colorblind even though he carries only one allele for color blindness. Yes, Alaki.
Sam is male.
So first of all, Sam is a male.
X and Y chromosomes.
So he has X and Y chromosome, yeah?
So he only has one chance. The allele will be passed on to him on the X chromosome. if I'm
Good,
not
he only
mistaken.
has one chance because if he has it on the X chromosome, there is no other X to have the dominant gene, whereas females have XX. So if they have the recessive one here, small c, they still have another chance of having a capital C to counteract that small recessive gene on the other side. Not small, it's a wrong word to use. The recessive gene on the other X. The male, however, only has one X. So if he has the small X, the Y cannot do what the other X of the female can do. Excellent. That's correct. We move on. Use a genetic diagram to determine the possible phenotypes and genotypes of children by a female with normal sight crossed with a colorblind male. I won't pass that on to you. Let's just look at the answer very quickly. If we're looking at this and they use small b for colorblind, you could have used C or and a homozygous normal there. And here's your story coming out there with this coming out. There's your gametes, and this is what you will get. XB, XB, and XB capital Y there. Remember that that's the only two that you can get. These are the two. This one and that one gives you this. This one and that one still gives you this. These two will only give you this. I'm hoping you're seeing what we're saying there. All right? Good. Let's go to meiosis. Name the organ in the human female where meiosis occurs. Where did you stop? Right?
In the ovaries.
Wow, Wasim knows about the ovary. Right, there's the ovary. Good. That's 1 point to that group.
That's our turn.
Hey?
That's our
All right,
turn.
we'll give you 2 turns then. Have you got any more questions?
Yes, we have.
All right,
It's-
there's a chance for you.
Okay, there's a question coming from Romeo.
Romeo?
Pahlela wanting to know.
Juliet.
It's
Romeo is asking where's
the
Juliet.
other
Okay.
way.
Yes, Romeo?
He wants to know, can speciations occur amongst the human species due to a geographical barrier and different environmental conditions?
Okay, that one that these guys won't be able to answer. His question wants to know whether speciation can occur during between humans because of a geographical barrier. First of all, let's talk what he's talking about. He's talking about allopatric speciation or geographic speciation. We can't say that because it's not been happening yet. According to scientists, they're seeing the speciation taking place in other animals. We haven't seen it taking place with us. However, if we go a little bit back and we talk about the different Homo species, there may be an explanation with the allopathic or other speciation that may have taken place over time and that. But remember, fortunately for you, that's not in the syllabus. You just need to be able to explain what allopathic speciation is with known examples and if they give you a question in that way. So, sorry guys, that one doesn't help. Any other questions?
No.
Sorry, we can't help you guys. Let's move on. Right, so we were here by you. Now we'll give you 2 questions. Identify the phase of meiosis in which the process represented in the diagram takes place. This thing here is taking place. Which phase of meiosis?
So phase 1.
Prophase 1, excellent. That's 1 point for you. And again, what process occurs at point X?
Crossing over.
Crossing over. So you got 2 easy points there. You covered up a little ground. Good. What is the significance of the process mentioned in 513 for 1 mark?
As I said earlier, variation in genetic
Variation is
material.
good enough. You get your point. Sorry to cut you, but it's as easy as it goes. Now we come back to you. And this one is worth 3 marks, John. I'm hoping you're listening to me. This one is worth 3 marks because it's a higher order question. The sex chromosomes are responsible for the fact that in human population, there are approximately as many men as there are women. Explain why this is true.
When you do a monohybrid cross
Yeah.
with X, Y, and XX, there's a 50% chance it will be a male and a 50% chance it will be a female.
There's a 50% chance of them being male and 50% chance. So you get your 3 points because of that. And we go back to you. Explain why color blindness, a sex-linked disease, is more often found in men. No, I can't give you that. You already answered that question. I'm sharp like that. We've already answered that question, mindsetters, that men, because they have only X and Y, they don't have a second chance. Whereas females, they have a chance on the second X chromosome to have the normal gene that will outdo the other one. Excellent. We're doing well, guys. Okay, I'm just going to then show you the question on genetics quickly. Sandy has given birth to a baby girl. Unfortunately, guys, no more questions for you. There are 2 men claiming to be the father of the child. Explain how the inheritance of blood groups and paternity tests could assist in establishing who the father is. Guys, this was an essay on genetics which nobody thought would come out, but there it is on there. And most of you can solve problems in this way, but you don't know how to explain it. So I'm just going to open the answer just to show you how many marks there were for you to get in this question. First of all, just by saying what controls blood groups, and then saying the different types of blood groups, and then giving an example, if this particular individual marries that particular individual, this is the possibilities of them getting that. That's the first part. The second part is about paternity testing, that means DNA testing. So if you just answered that, you would have got at least 10 to 12 marks in this essay. It wasn't difficult. Look at the answer at school and work from that. With that, guys, I'm hoping you enjoyed what we've done. I hand you over to Pumi, who's going to wrap it up for us.
Oh, thank you so much, Aslam. Guys, there is one another interesting thing that I want to tell you about right there on our Facebook page. So make sure that you comment on the picture that has been posted on the Safe Way Facebook page, and then you can get to win yourself 150 Google Play points. So in order for you to win yourself that, please comment on that page. And also, we have a question that we asked you earlier on today, which is, what are your views on people that buy license? You know, if you buy a license, or you know, if somebody who's bought a license before, please make sure that you comment right there so that we can make another video and post it and see us interpreting all of that on the page. Right now, guys, I have to love and leave you, but before we
Give
do
you a
that,
loud
can we
shout-out
get
quickly.
a quick bye-bye from you guys?
Bye.
Bye, guys. Thank you so much for
Bye.
coming.
Bye.
Awesome stuff. It has been an awesome show. Thank you so much for joining us. This is, of course, the last week of the live shows. See you next week.
Keep studying and vote for me. Thanks, guys.